Where the axon terminal of one neuron interfaces with the dendrite of the next neuron is a 20 nanometer space called the synapse. The axon terminal is also known as the synaptic terminal. A neuromuscular junction, shown here with my mouse, is the synaptic space, or cleft, between the axon terminal of a neuron and a muscle cell. So the synapse is the 20 nanometer space between the axon terminal of one neuron and the dendrite of the next. To continue the impulse from the presynaptic neuron to the postsynaptic neuron, or effector, requires neurotransmitters. Neurotransmitters are released from the presynaptic membrane, diffuse across the synaptic cleft, and bind to protein receptors on the postsynaptic membrane. These chemical messengers are released at the synaptic terminal from vesicles by exocytosis when the action potential reaches the axon terminal. In less than 1 millisecond, the neurotransmitters are across the synaptic cleft and start binding to the protein receptors on the postsynaptic dendrite. Excitatory neurotransmitters cause neighboring sodium ion channels to open, causing local depolarization, which, if the threshold is breached, turns into an action potential, which moves along the axons down to the axon terminal, then to the next neuron or effector. Inhibitory neurotransmitters cause potassium ion channels to open, hyperpolarizing the membrane, making the threshold more difficult to reach. A single cell body could be integrating the signals from many presynaptic neurons at the same time. The cumulative effect, called summation, determines whether an action potential follows or not. Neurotransmitters can carry the signal from a neuron to an effector, gland, or muscle. The image on the left is an electron micrographic picture showing the axon terminal of one neuron interfacing with the effector cells, in this case muscle cells. The fate of neurotransmitters. Neurotransmitters can't stay in the cleft. Overstimulation of the postsynaptic membrane by excitatory neurotransmitters keeps the membrane in a constant depolarized state. They need removing from the cleft, and quickly. Obviously, many would bind to the postsynaptic membrane. Most that don't are consumed by synaptic enzymes specific to that neurotransmitter. Acetylcholine is the excitatory neurotransmitter for muscle contraction, whereas cholinesterase, sometimes called acetylcholinesterase, is the synaptic enzyme that breaks down acetylcholine. MAO, monoamine oxidase, is a synaptic enzyme that breaks down dopamine, an excitatory neurotransmitter, in the pleasure center of the brain. A chemical in cigarettes blocks this enzyme, keeping more dopamine available in the synapse. Tetrodotoxin, found in skin, ovaries, liver, and the intestines of the pufferfish, fugu in Japan, blocks sodium ion channels by preventing acetylcholine attaching to postsynaptic membrane receptors. Batrachotoxin, secreted by the skin of a Colombian poison arrow frog, is the most potent known venom. It acts by increasing sodium ion channel activity. An antidote to pufferfish toxin, perhaps. Some neurotransmitters do a 180 and head back to the axon terminal of the presynaptic membrane, attaching to protein receptors there before being taken in. Serotonin is a neurotransmitter that regulates body temperature, sensory perception, and mood. Too little causes depression. Too much makes you sleepy. The individual on the left here shows the synaptic cleft with a normal amount of serotonin. The presynaptic membrane shows an appropriate number of protein receptors. The protein receptors in the postsynaptic membrane are not shown. But on the right there, we see that there is an individual who has many protein receptors on the presynaptic membrane, which means too much serotonin is being taken up by the presynaptic membrane, not leaving enough in the synaptic cleft. What SSRIs do, drugs that actually selectively inhibit reuptake, competitively bind to the protein receptor on the presynaptic membrane, limiting the amount of uptake into that presynaptic cleft. This drug is Ritalin. It prevents the reuptake of dopamine. Cocaine not only prevents reuptake of dopamine, but also increases its release. The other fate of the neurotransmitters is that they diffuse out of the synaptic cleft altogether. Of special note are the endorphins, neurotransmitters that act as painkillers in the synapses of the brain, and also affects the emotional centers of the brain. Curiously, a deficiency has been linked to alcoholism.